All righty, folks, we're going to go ahead and get started. Thank you all for joining us today, and those of us online as well. It's my pleasure to introduce Dr. Nathan Fumia. Nathan's had a long storied career so far. Prior to grad school, he was a PCA, cannabis greenhouse operator, and a fourth generation citrus and almond farmer. Pistachio, I apologize. In California. After that, he moved on to graduate school at the University of Hawaii. For the last couple of years, he's worked with the World Vegetable Research Center. He's worked with the Hawaiian Agricultural Research Center, HARC, and we'll get to listen to a very exciting presentation today on all that research. So thank you very much. Please welcome Dr. Fumia. And if you would like a business card, we do have copies.
All right. Thank you, Albert, for the introduction. Thank you, everybody, for coming who's here in person, and those folks that are here on Zoom as well. So today I'm actually going to focus a little bit on one of my chapters from my PhD dissertation. This would be the fourth chapter where I did stochastic simulations, looking at neodomestication, so how we can do population improvement in wild and semi-wild species. And then I'll be providing some of the insights that I gleaned from those simulations and how I've integrated them into my thought processes and new and old plant breeding projects that I have ongoing at the Hawaii Agriculture Research Center. And so, like I said, I want to introduce first the organization that I work for. Albert did pretty good at my personal background, so I'll quickly go through that. And then the stochastic simulation, I'll introduce some of my thoughts on both historical domestication, neo-domestication, and build the framework that we could start from. And then the methods that I apply in order to set up breeding scheme development. And then some of the results that are from that and some of the conclusions and insights. And then we'll look at that and apply it to some of the ongoing breeding programs that I have at the Hawaii Agriculture Research Center in coffee, moringa, and pineapple. So who is HARC-HSPA? So the reason I put -HSPA is because we are an organization that started in 1895 as the Hawaii Sugar Planters Association. So we're a trade organization based in Oahu, but had experiment stations and cooperating plantations across all of the Hawaiian Islands. And we did everything from agronomic and plant breeding and genetics research to chemistry, as well as engineering and milling. It was about 100 years later that the plantations started to see the writing on the wall and began to close their doors, either due to high labor costs or different issues, water availability also being one. And so HSPA then began to diversify some of the research that we were doing at that time to help the plantations be able to shift to different crops. And it wasn't until, I think, 2006, roughly, that we shifted to the Hawaii Agriculture Research Center, where our primary mission is to do diversified agriculture research to benefit both local farmers in the state of Hawaii, but also in the greater subtropics and tropics. And so we have two research stations on the island of Oahu. The first is Kunia Research Station, which is a 100-acre farm. Where we have year-round crop production, winter annual temperatures of 78 degrees. So I know you guys were saying 55. Yesterday was warm for you, but for me, that's still pretty cold. We also at this site have laboratory facilities that were built in the, I believe, roughly 2008, where we have a full micropropagation, tissue culture, transformation, DNA extraction, and amplification lab. And we also have an education center there where we bring in K through 12 students and do outreach activities to the community as well. And we have about 500 students a year that come through this education center, both for daily field trips as well as summer-long internships. And this is an irrigated site, so we only receive about 15 inches of rain per year. So it's a relatively dry site. The other site, the Monowili Breeding Station, this was historically the Hawaii Sugar Planter Association's breeding site for sugarcane. It's a very unique microclimate. And this receives about 180 inches of rain a year. And on the other side of the island. So it's a windward, so it receives all the trade winds and gets a lot of rain. So we don't irrigate here. And we primarily focus in perennial tree crops here. So that being cacao, coffee, as well as some endemic hardwood tree species that we work with. And so our project leader staff include individuals with, a variable level of experience, both throughout basic and applied plants and environmental sciences. So they have different expertise, and we collaborate on variable projects depending on what we're investigating. So first, I'll just start off with Mingli Wang. She's a molecular biologist by trade, and our micropropagation and transformation laboratory manager. Tyler Jones is a horticulturalist and manages the Monwilly breeding station. Nicholas Dudley is a senior forester and does some breeding of native endemic hardwood tree species, such as Acacia koa, some native sandalwood species as well. And he works very closely with the restoration and conservation industries in Hawaii. Johanny Rivera-Zayas is an agronomist and has experience in soil and environmental sciences. Maureen Fitch is a plant physiologist who was part of the team that developed the gene-edited papaya back in the 80s. Julie Burden leads our agrophotovoltaics projects, where we're testing different crops and different varieties underneath solar panels. And Linda He is a molecular biologist. And so we have several research scientists and technician staff that we share amongst ourselves, depending on what tasks are at hand. But we have roughly 10 field technician staff and 6 technician or scientist level staff within the laboratory. And they'll help with everything from micropropagation transformation to agronomy and field operations and forestry work. And so HARC also has a long history, as I had mentioned earlier. So we have a lot of ex-HARC staff that we regularly collaborate with, depending on if we need assistance with tropical genomics. We have Dr. Ray Ming, who is at the University of Illinois, and then now at FAFU in China. We have an agronomist emeritus who has decades of experience as well that we can reach out to. So I'll now just hop into a little bit of my personal background. Albert did a pretty good job, but I grew up in an orchard management company in California. We have about 1,000 acres of citrus and pistachios, and I learned a lot of the operations from a young age, both in managing the orchards, but also in dealing with export markets. I went to California Polytechnic State University in San Luis Obispo and studied agricultural business and pest management, really. And so I became a crop consultant after that in Watsonville and Hollister regions. So I worked in dozens of specialty row crops, both focusing in agronomics, but also IPM and irrigation management, things of that nature. And after that, I shifted into greenhouse management of a cannabis facility in San Diego. I did that for 2 years, and we used technologically advanced systems, so sensor-based environmental controls, as well as integrating camera vision along the tops of the plants for assistance with scouting and adjusting irrigation regimens and fertility management. And so this whole process kind of is what led me to go back into school and study plant breeding. I saw throughout this the potential yields of crops, or the potential quality of crops, is always limited by the genetics that you were working with. And so that seems like the last frontier for me to better understand that. So with all that background, this is the paper that I'll be discussing for the next probably 20 to 30 minutes, hopefully 20, which was the 4th chapter of my dissertation. This is more theoretical in basis, but it is based on the 3 chapters that were previously done, where I looked at the breeding cycle with specific crops that we have at the Hawaii Agriculture Research Center to better understand parameters that are involved in crossing, evaluation, or selection. And so a lot of the inputs and assumptions that I make are based upon those 3 chapters. And I'd be happy to share the full dissertation with anybody who's interested in reading that. So we can first start off with historical domestication. So historical domestication can just be defined as human-mediated evolution to adapt plants and animals to whatever our human preference might be, right? So this has been done to improve plants for increased productivity, harmonize crop management practices, so agronomic performance. It could be promoting the ease of harvest or consumption, or even improving the palatability of crops. And it's a result, historical domestication was a result of conscious and unconscious selection. So a lot of times things were selected and we didn't know what we were selecting for, but it was occurring and it was shifting the genetics of those populations. So the neo-domestication is very similar to historical domestication. There's just a couple major differences, right? We now know what we're doing and we're leveraging conscious selection by using modern techniques and technologies to do so. And the traits that we're still selecting are still more or less the same. There's still those that satisfy human cultivation and related activities, and we're trying to fit into a human market preference, right? And so typically newly domesticating a species, we're using wild species to fit into that market. But we can also look into the broad genetic base that might be available in semi-domesticated crops as well when we're doing population improvement to fill in these niches. So we can consider orphan crops. So like this ground cherry here, there's been some work in ground cherry using gene editing based on its relationships with tomato. And so these are typically locally critical crop species. And so they've undergone minimal local selection. And so they harbor some of those human preferences that we could then expand to larger production regions. We could also look into landrace cultivars as well to improve the diversity of our food systems. And so we can transition these by leveraging advanced rapid breeding techniques. And understanding how we have all of these species that are typically undercultivated at the global level could improve the security of our food system at a regional or local level, which is something that I'm particularly interested in. So when we think about domestication, we can look back to Dr. Harlan from the 70s, who defined the domestication syndrome. He primarily focused on annuals. So with the list I have here, ranges from simple to complex genetic architecture and of what would be considered a domestication syndrome for an annual grain. But if we think about vines, tubers, or tree crops, that is a very different situation. And oftentimes we're not seeding those crops, we're selecting clonal propagates, either grafting them into the field or not. So the variations that we might want within that the genetic architecture for that specific trait would change depending upon that market that we're trying to fill. And so we can look at this pyramid that I created and begin to understand how we have a trade-off between genetic diversity and agronomic performance. So wild species, so within this, we're typically conferring genetic diversity, or sorry, we're decreasing genetic diversity, and that's conferring an increase in agronomic performance. So as we move up this pyramid, we're selecting variants, and that swept variation is then occurring during this bottleneck of population improvement, and then we're removing that segregating variation from our populations. And so the three groups that I'll be talking about later on, terming them as wild, orphan, and landrace, but we can think about them on this type of a spectrum where we have a decrease in the background genetic diversity and, in theory, a higher agronomic performance. So we're having different potential germplasm acquisition scenarios. And so doing that, just orphan crops, typically thinking about how they have a little bit more genetic diversity than land races because they had undergone maybe a shorter or broader domestication bottleneck due to their regional importance or a lack of recent research funding. And so we can also think about neo-domestication as occurring along this continuum, right? So where sometimes we have simple traits that we want fixed within our population. These are typically cultivation or breeding related traits that need to be fixed for us to be able to operate. And then you have some more complex traits that might need to retain enough genetic variation in order for you to make sustainable gains across the cycles of selection. And no two species are the same in terms of what their syndrome traits might be. And so we can see this as a kind of continuum of domestication. And so I do want to also mention at this point that this doesn't even consider market. So when you're doing a neo-domestication project, it's important, in my opinion, to consider the market of which that crop will actually enter into. We can achieve substantial gains over the wild type. You could say 200, 300% over seed size of the wild type. But if you're entering into market competition with a crop that has a seed size that you're still only a fraction of, I don't think you're going to have very many farmers adopting that new crop. So understanding the endpoint as well is important. And so first, we're going to start off with the breeding cycle. This is kind of the first step of understanding breeding scheme development. And so the breeding cycle is, at least for, the breeding cycle is so crossing, evaluation, and selection. And so we have different parameters that are involved within each of these components and each of these steps. So within crossing, we can decide on different parameters, such as the number of parents we use, the number of crosses, the number of progeny, what type of the cross, are we using mate allocation. Evaluation, we can use more locations, fewer locations, higher replication or lower. We can use more checks. We can have a better experimental design for precision of our estimates or not. And we can also leverage some subsampling to increase those estimates. And then selection, it's that component, again, that really differentiates neo-domestication and historical domestication. And so you can select the percentage of individuals, selection intensity. You can use a different selection methods. You can use culling and truncation, or you can select on an index. You can select on a different unit, whether it's families, lines, or parents, or a different criteria. You can select on the phenotype, the genotype, breeding values. And so each of these parameters creates a different result in the gain that your population realizes over generations. And so I'm sure this is a formula at least most of you have seen before. So I'll run through it rather quickly, but this is the breeder's equation, genetic gain equation, and this is a way for us to track population improvements and also predict population improvements. And so we have that genetic gain equation being a combination of the selection intensity times the additive genetic variation times the selection accuracy over these cycles of selection. And when we're looking at these simulations, the best way to track that is through the additive genetic variation. And we get to that because the variation of the phenotype is equal to the variance of the genotype times the environment times their interaction. And the variance of the genotype can be broken down into additive, dominance, and epistatic control. And so this additive component of variation provides the basis where we can look at genetic variation and we can define if something has been fixed within our population or if it has the potential for future gain and how much gain that's possible for that given trait. Sorry, I didn't keep clicking there. And so genetic gain can be improved through different specific focus in these different parameters within the breeding cycle. So selection intensity can be increased by increasing the number of selection candidates that we have while holding those selected constant. Oh, apologies. We can increase the additive genetic variation by using things such as mate allocation. So we might be forfeiting some gain in order to reduce the inbreeding of our population. In selection accuracy, we can increase primarily through increasing the heritability for the trait that we're interested in. And the number of cycles we could increase either through predictive methodologies like genomic selection, getting multiple generations within a year, or increasing plant development through something like speed breeding, where we can get more generations within a given year. And so that interaction between breeding cycle components and the breeder's equation is what's driving the simulation I'm going to present. And so it kind of provides that theoretical background where we can track population improvement over generations of recurrent selection. So before breeding scheme development, which is the second step, I first needed to create some of the populations that we're going to feed into this. And we can think back to the wild, orphan, and landrace as being kind of these terms that we'll use. but at the end of the day, we're creating different variation for certain types of traits. And so to do so, I used AlphaSim R to generate different haplotypes by certain set effective population sizes and forming populations of different sizes. So creating this kind of germplasm acquisition scenario. Then I added two traits, one being a relatively simple kind of cultivation constraining trait, and one being more complex that could either be related to yields or adaptive fitness shift or environmental plasticity of the population. And this is something that we might want to maintain enough variation to be able to consistently shift our population to changing market preferences or changing production. And then I did differential recurrent selection. So for wild, it was just random selection of individuals across 40 cycles of selection. Or orphan crops was a more lenient, low selection intensity, and then land races being more stringent to kind of create these different starting points. And within this simulation, there's genetic drift occurring, there's mutation occurring, and this selection, we're trying to, again, generate these new population genetic dynamics that fill into this next step. So these, then you have a single population. So with that previous burn-in, ran about 20,000 different combinations of these parameters. And then they feed into this breeding scheme development phase, where then we're now introducing, again, more combinations of parameters. And so we're trying to, again, identify the optimal breeding schemes that would depend on a given population, wild, orphan, landrace, and that standing variation that's present within that population. So we can, again, try to have a high gain and a low additive genetic variation. So we're fixing that simple trait within our population, or we're having a high gain and a high additive variation in that complex trait, so we can begin to shift that population if we need to. And we're changing things, again, along that crossing evaluation and selection regime. And then after this, the simulations were run. I actually integrated different costs that I used from the different budgets from the breeding programs that go on at the Hawaii Agricultural Research Center, and use them for a field type crop, a horticultural crop, and a forestry type crop in order to get a range of costs that are associated, particularly with the variable costs. So the cost of genotyping a sample or the cost of phenotyping a plot. And then I use it as integrating into kind of a return on investment type of a framework, where you're looking at the final value of the investment, or the final genetic value, or the final genetic variation divided by the initial value, or minus the difference between the final and the initial divided by the initial. And then this kind of gives us an idea of how well our costs are being utilized for these changes. And so within these simulations, this is colored across 80 cycles of selection. The first 40, where you see that vertical line, is that burn-in phase, where we generated these different potential germplasm acquisition scenarios and these wild, landrace, and orphan type crops. And we have the simple trait on the top and the complex trait on the bottom. The reason I am reporting phenotypic value and phenotypic variance is, more often than not, we do not have genomic resources and emerging crops. And so this is much easier for me to compare against my current breeding projects, as well as I think some emerging ones as well. If you have access to that, that's great. And the reason we see these weird separations in phenotypic variance is because there were different heritabilities that were used. And so you're seeing that separation there in those lines. But this is what I was hoping to see, is that at that germplasm acquisition, at the end of the burn-in phase, that we have a variable level of the phenotypic value and a variable level of the phenotypic variance. So we have these different situations, we can begin to parse effects of these different parameters. Again, when I integrated costs, these were based upon existing breeding projects and a field type crop, which is Stevia rebaudiana, and a horticultural crop, which is cacao, and then a forestry crop, which is Acacia koa, a hardwood tree species. And I used the medium as representing what the budget actually was for those crops, and then going a little bit lower and a little bit higher in order to create these different situations. Those numbers were then multiplied by the total progeny that would exist within our population at a given generation. which is based on the number of parents, crosses, and progeny per cross. So in order to understand the effects of these different parameters, I used a multivariate mixed model where we have y as being the response, so it's the change in that target. So it's either the gain of the simple trait over the cost to attain that gain, or it could be the change in the variation of the complex trait over the cost that was incurred during that change in variation. X just being the design matrix for the fixed variables, so the population type that was used, the selection criteria, the heritability, the index weighting, and more. Beta just connecting as a column vector those fixed effects coefficients. Z is the design matrix for different random variables. So I wanted the number of parents, and the number of crosses, and the number of progeny per cross, as well as the effective population size and things like that at the starting point to be variables, so we can understand how that might change. And mu being obviously the vector of random effects, and epsilon just being our residuals. And so this is going to be a lot of colors and a lot of things going on, but I think it's just important to see how it changes as we move from a more cheap, less resource intensive crop like a field crop into horticultural, which is more resource intensive, into forestry, which is extremely resource intensive for phenotyping. And we can see the differences in terms of the importance of different covariates right there on the left, and for the different types of targets that we have. And we can see that different covariates become more statistically significant in terms of having a change in the target. So then some of the parameter effects here, just reporting heritability and index weighting as the jitter and the box plots. On the bottom x-axis, we have 9 groups. The first 3 are landrace, the next 3 are orphan, the last 3 are wild. And within each of those groups, it goes phenotypic recurrent selection, maximum avoidance selection, and then genomic selection. Genomic selection is using GBLUP, basing on genomic relationship matrices. Maximum avoidance is based on pedigree relationships, where we're trying to reduce the amount of inbreeding. So we're using optimum contributions selection, but instead of instead of crossing those individuals, it used what is circular mating in order to keep my populations the same size throughout the simulation. The reason we don't see a lot of change in these phenotypic value and phenotypic variance on that simple trait within the landrace and the orphan is because it's exactly what we would expect had we acquired germplasm from those types of populations, where that simple trait likely would have already been fixed due to local and regional selection. So this fit exactly what I was hoping it would, which was a good check. And then we can see very, very large changes in the wild. And then the structure of these graphs is the same. Now for the complex trait, we can see variable changes, and variable changes in the target by that cost for different heritability and index weightings on the trait. And so this was important to understand that there are differences in terms of how we employ these parameters, in terms of the gain that we can achieve, or the changes in the variance that might occur during population improvement. Because we have these 2 targets that we want to look at, we have a simple trait that we want to have fixed within our population, or we have a complex trait that we want to maintain variation over generations, and we don't want this to fix. I employed asymptote discovery, where for every the tens of thousands of simulations that were run, I tried to identify where the asymptote and where the sigmoidal curve approached that asymptote to see how many cycles it takes for those different traits. And so this is just done through nonlinear least squares, where y is the change in that target per dollar spent within the breeding cycle. D is the maximum phenotypic value for that given scheme, that given set of parameters that were employed within the simulation. A is the minimum phenotypic value or variance for that given scheme. C is the point of inflection, or the mean phenotypic value. So it's the point where the sigmoidal curve changes. A was already explained. X is the cycle that we're trying to discover within this formula. And B is just the hill slope of the curve that's used within this type of non-linearly squares. And so we can look at a single case where we have a simulation and we're looking at the phenotypic variance for a simple trait that we're trying to fix. And with a heritability of 0.7, I randomly selected throughout the simulations for examples. So we had a heritability for that simple trait of 0.7. We employed genomic selection. Maybe we chose a wild species that we have a related species and we have genomic resources for it. And we can see that when we fit the sigmoidal curve, it fits that simulation very well. And we can identify the asymptote of where that variation is completely lost, where that curve approaches that asymptote, which was 6 generations of breeding. So for genomic selection, because I was training my populations every other generation, this is 3 years. You can also think about a complex trait where we're trying to maintain gain over many, many generations. I wanted to look a little bit more at the maximum avoidance because I'm particularly interested in, especially in the tropics, being able to shift populations as I need to, given how variable things can occur, especially in Hawaii. And so selected something that had a 0.3 heritability for a complex trait, which I think is more or less pretty reasonable, what you would expect. And use maximum avoidance. Again, that's optimum contributions. We're trying to reduce inbreeding. So we're selecting parents that are not related to one another, or at least less related to one another than you might select otherwise, and using circular mating with that. And it was from an orphan type population who can fit that sigmoidal curve and find that we don't have that curve approach the asymptote within the 40 cycles. So there's plenty more variation and plenty more gain to be achieved within that population past 40 cycles of selection, which in the tropics and a lot of the crops I work with is well beyond my lifespan. So that was good to see. So some of the conclusions that came out of this was the first major one, where in all schemes and all populations and each of those cropping systems, so those variations in costs, the improvement in gain on the simple trait and the reduction in variance, or the improvement in gain in the complex trait and maintenance of the variance in that complex trait, was always improved and increased had we started with a large population and a large effective population size of that starting population. And I'll bring this slide back in a few slides. If we're trying to fix a simple cultivation constraining trait, it was kind of a middle level of parents and a low number of progeny per cross, having a high heritability for that trait and a high index weighting, and just using phenotypic recurrent selection. By doing this, we're fixing that simple trait within our population, so we're reducing our additive genetic variation, and we're highly accurate with our selection with that high heritability and high index weighting. If we're trying to make rapid and cost-effective gain in a complex trait in these wild and semi-wild species, then having a large number of parents and a large number of progeny per cross with a high heritability and using genomic selection where we're able to, as well as a high index weighting, we're able to make that rapid and cost-effective gain. And that's going through different changes within a higher selection intensity, where we are sacrificing and losing that additive genetic variation there. We're highly accurate because now we're selecting on the genotype, the genomic selection. And we are increasing the number of cycles per year by being able to go through 2 generations within that year using genomic selection. And then if we want to be cost effective in the gain and the variation that we maintain for a complex trait, then it was a high level of parents and a lower number of progeny per cross than we previously saw on that slide. And we're actually okay with a lower heritability of that trait. We can use maximum avoidance selection and not weight that trait as heavily. And I'll explain a little bit more on the next slide why that might be an okay situation. And there we're kind of sacrificing selection accuracy in order to maintain some of that additive genetic variation. And we're resulting in less gain, but we're maintaining more variation in our population. And so some of these implications, I mentioned it earlier, we have a potential species that we want to domesticate and introduce into production onto farmers' fields. And sometimes you can identify a market niche, and that's the ideal situation. And if there's no cultivation constraining trait, then in reality, you can shift right into production. And you can begin just breeding for the harvestable organ. If you have a constraining trait, you'd likely want to fix that within your population if it was something like seed shattering or a branching trait you didn't like. And then you can begin breeding for the harvestable organ. If you enter into a situation where you're trying to turn to a market where you're going to have competition, then you can't really enter into production at any reliable size until you've bred for that cultivation constraining trait and the harvestable organ, where you can actually meet the competition that you're trying to enter into. Okay, so we'll try to take a second. I'll sip some coffee really fast. So this figure right here is really how I've based a lot of this finding, and this figure is how I based a lot of my current ongoing work at HARC off of, where it was extremely apparent and makes a lot of sense that the larger the population that we have and the higher effective population size that we have of that population is going to improve us in all situations. And it's doing that because we can have enough variation to select specific market types for our crop, so we can try and identify a market niche that might fit into, and you're going to have the variation there. You also have the ability to identify unique and novel variants within that population, the germplasm population, where oftentimes these agronomically beneficial or agroecosystem beneficial traits are extremely novel within wild species. And we can also maintain enough diversity within our germplasm in order to potentially perform genetic rescue for our populations if we begin to succumb from inbreeding depression. So we have a lot of these benefits that come from just having those large populations. And so the first case that I'm going to start about, and these are going to be relatively brief. Again, I've only been with HARC for about 15, 16 months. So a couple of these are rather new. So I'm going to present on kind of what I've done thus far and what my near-term and long-term plans are. But that's about as best as I can do at this point. So for moringa breeding, I started this last year, and this is in cooperation with the World Vegetable Center out of Taiwan, where I have an ongoing collaborative working agreement with, and I imported their international moringa collection. Moringa is a leguminous tree crop that has high protein content in the leaf as well as in the beans, and it's extremely drought resistant as well. So it has the potential to fit into a lot of food secure niches. And even more beneficial is there's 4 very clear markets, and some are regional and some are international. Some of the regional ones are different populations will consume the leaf as a fresh leaf in soups and things of that nature. But on the international stage, it's become extremely useful due to a lot of the vitamin contents that are present within the leaves to actually grow the crop, harvest the leaves, dry them, powderize them, and turn them into supplements. And this is roughly a $10 billion market currently. There's also some populations that consume the immature beans, somewhat like you would consume string beans. And you can see some of the variation. I hope that's clear enough. It's trying to get a good picture. Some of the variation in the color of this germplasm in terms of the juvenile beans. And then there's also the mature beans that you can utilize either for cosmetic oils because of the high oil contents, or for protein because of the high protein contents in those mature seeds. So that's 4 very clear markets. So currently I'm in the stage of germplasm characterization. So I have types from South and Southeast Asia, Polynesia, and East Africa that are growing in the field together. And my plan is to separate these into market-specific type populations based upon the traits that fit those markets best, and to integrate technology because, again, these are tree crops, and I don't know if you can tell in that picture, but I'm standing on top of my car, and these are only 9 months old, so they can grow about 4 meters in 9 months. I have some dwarf types in there that I'm sure I'll be able to phenotype with relative ease for a couple of years, but some are already 10 feet tall. And so I'm not really trying to carry a ladder around fields in order to do that. So the plan is working with World Veg, as well as Dr. Ray Ming, who I had mentioned earlier, who has a lot of experience in tropical genomics, to put together a pan-genome on this population. And then to also integrate proximal sensing. So I've been collecting NIRS reflectance data and plan to be sending out samples for metabolomic analyses, so I can get some secondary traits I can kind of reduce my costs on in order to value different germplasm. And so another one is reinvigorating what was once the HARC pineapple breeding program. This started in 2003 and ended in 2015. Luckily, our field crew has been around for long enough that they kept the field alive, which had a ton of F2 individuals with some wacky fruit shapes, some really interesting aromas, and some really interesting flavors. And this was a kind of a cross primarily with the goal of understanding evolution of pineapple, but it created a lot of unique variation. So the F2 population is segregating, and I say pseudo F2 individuals. So most pineapple is outcrossing, so you don't really have true F2s if you have an outcrossing individual, so they're pseudos, but they're still segregating for the traits of interest. And they're segregating primarily from the Bracteatus genetics for self-compatibility, as well as different leaf, and obviously, as you can see, fruit morphologies. And within this population too, there's some individuals that taste like coconut. And so what I began thinking about was, okay, well, let's bring over the USDA collection from Hilo and set up a new field. So we ripped out the old field and planted it with the USDA collection. And some of the quality agronomic traits that I'm interested in are with that coconut flavor F2 individual, to maybe create a pina colada pineapple, because there's some white fleshed pineapples that are within that collection. But to do this characterization across a lot of the variants that exist in Anonas comosus, as well as bringing in some related genuses. I'll explain why I have those related genuses in a short second. Again, we're integrating technologies here where we're going to be putting together a pan-genome to understand the diversity of the collection, as well as to begin to possibly set up markers that are associated with certain traits of interest, and then also to introduce proximal sensing. And primarily going to be selecting for quality and agronomic traits. And some of these, quote, long, no clue if they're going to work, so I'll just start off by saying that. But we have some self-compatible individuals in our pseudo F2 populations. And I spoke with my predecessor, who had said some of the individuals at the F1 were fertile and some were sterile. And that sterility maybe occurred at the F1 and maybe it happened at the F2. And so it seems to be occurring at these different stages. And so to be able to breed a type for true seed pineapple instead of having to do clonal propagates could change the pineapple industry pretty dramatically. And then another long shot trait, and again, this one is, when I say long shot, Pitcairnea gracilis and Pitcairnea puniceae are C4 photosynthesizers. All of pineapple is a CAM photosynthesizer. My thought is if I can get these to cross reliably and generate a C4 pineapple so that we can have a 1-year crop potentially instead of a 2-year crop, again, it could dramatically change the pineapple industry. So that's kind of how I'm looking at that. And so both of those situations were me trying to get as much genetic diversity as I possibly can and as large of a population as I possibly can within the resource constraints that I have at HARC. And trying to integrate technologies in order to reduce some of the costs that I have in terms of phenotyping or being able to select. And then the last is the HARC coffee breeding program. It's gone on for much longer than 1999, but 1999 was when the first crosses were made by Dr. Shifumi Nagai. And this was primarily for coffee leaf rust resistance, pulled in from Coffea robusta into Coffea arabica, which is the probably 99.9% of production in Hawaii is Arabica coffee. And so we have about 5,000 F2 individuals within that population. Coffee is a slow crop, and we also had a lot of pushback from farmers where they wanted to They did not want any Robusta in their types. And it wasn't until 2020 when coffee leaf rust entered into Hawaii that they wanted a disease-resistant type. And luckily, Shifumi Nagai had made those crosses. But they weren't happy about it until 2020. So sometimes the farmers are helpful and sometimes they get in the way. And so I think using some of that foresight is important just in terms of knowing what is coming down the line. And so we have 5,000 F2 individuals. We have feral coffee from a plantation that was before HSPA was in Manawili Valley. So we have a bunch of feral coffee that's just growing wild out there and a lot of spores that we can collect, and we can scrape off those spores, get counts, inoculate our F2 individuals. So we're doing a bioassay here, and then we evaluate them after about 5-6 weeks and see if we see spore growth, which and the bottom one you can't really see too clearly, but the top one you definitely can. So we're removing those individuals, and we're now at the stage where we're working with our farming partners, where we have these varieties pushed out into these farmer partners on all of the major islands of Hawaii. So Hawaii Islands, Maui, Oahu, and Kauai, in order to evaluate them for agronomic traits as well as quality traits. So we can get the farmers to be secure in understanding that if they adopt these disease-resistant types, they'll also have high quality. And so I'd introduced Julie Burden earlier on. She does lead our agrophotovoltaics, but she is also a specialty coffee Q grader. And we also have, because Shifumi Nagai is from Japan, she has a very good relationship with Suntory, who has developed a lot of metabolomic analyses for coffee and coffee quality in particular. So we're working with them in order to present to the coffee farmers in the state of Hawaii that these varieties are in fact going to taste better and you won't have to spray fungicides all the time. So with that being said, I didn't put in an acknowledgement slide because I think I don't know if I would have been able to fit it in a single slide. So I just want to say that anybody who has been a part of my pursuit towards my PhD and after part of these different projects, that science is not done in a chasm and by yourself. And I appreciate every one of them. So now I can take some questions if y'all have any.
Welcome. Any questions? We do not have a microphone, so please do raise your voice. And if you could repeat the question, Nathan, once you get it.
Yes.
What's your biggest piece of advice for juggling multiple different crop species, like the diversity of just species that you're working with?
Yeah.
And also kind of honing in on what your breeding targets are. Like I guess what's your wisdom after?
Okay,
after
so the question, so the question is, how do you manage both a lot of different species and as well as what the breeding targets are for those species?
Yeah, just the diversity of objectives.
I'm being honest, I'm still figuring that part out. I've carefully selected things that I either have regular flowering, because a lot of times the constraint in these, I'm going to be having to make hand crosses because they're not wind pollinated. So moringa will flower for 9 months roughly. So that gives me a large window where I can make crosses, so it's flexible. Pineapple is primarily driven by hormone sprays, so I can schedule that accordingly. I have some private contracts that I didn't even present in here, but there's a few species that will, again, either flower regularly or they're outcrossing species, and I can plant them into separating crossing blocks in order to make specific crosses and take leverage of bees and things of that nature. So to answer that question is being a little anal about organization is probably how I've survived. And then in terms of breeding traits is doing market research and either looking at commodity reports and things of that nature to better understand both what farmers and consumers are valuing, and then looking at how that might be forecasted to better understand what could be a good trait to focus on. Yeah. And then others like those long shot traits in pineapple are just out of general curiosity. Yeah. Yes.
I may have missed this, but do you have Did you say you have farmer partners for the moringa work at this stage?
I do not. I'm currently working with the World Vegetable Center in Taiwan, which has sites across Southeast Asia as well as Eastern Africa. So my hope is later on, probably once I can get reliable composite populations to work with them in order to push them out into those regions and see the receptivity. There are some local farmers in Hawaii as well, given our large Asian and Polynesian population. So to work with them closely as well. But yeah, that's a little bit further down the line currently. Yeah.
And so my other question about the moringa is you said that there is already a market, sort of.
Huge market.
A huge market
Yeah.
for the leaf extract,
Yeah.
like the.
The powderized supplement, yeah.
So how long, on average, do the current, varieties or
Mhm.
types, land races,
Yeah.
how long can you continue to harvest leaf? Like, what's the life of the
Yeah,
tree?
so there's evidence that you could, and they regularly retune or grow when you cut them back. So there's a farmer in Hawaii who will harvest moringa until it's above what they can actually pick, and they'll cut them back and they'll retune and grow. And those trees, by my estimates, are probably 20 years old, roughly. And there's some very advanced mechanized harvesting that's going on in Israel as well, for moringa, which is high density planting and similar type of a mechanism. For that market, I'm particularly interested in nutritional composition, so increasing nutritional composition in a variety, as well as increasing leaf size, because a lot of these varieties have very small leaves. But there's variation. I don't know if you remember the picture where one of the leaves probably covered, I don't know, 25% of my hand. And that was just a single leaflet. So there's potential ways to create a higher yielding or a higher quality variety. Yeah. Yes.
What are the financial sources to support this work?
Yeah, good question. So for moringa and pineapple, we have a long-standing relationship with the USDA out of Hilo. But it's pretty much through ARS, which is the acronym is SIMBRU. I believe it's the Center for Molecular Biological Research Unit, or something along those lines, where we receive yearly funding based on 5-year contracts from them in order to do preliminary work for research in the tropics. So that, and then the holder of that within HARC, we then submit internal proposals to in order to receive funding. But that funding has been coming to HARC since 1978, I believe. So it's rather regular. And then because HARC is a 501c3 nonprofit contract research organization, we have, I have several private contracts for custom breeding for companies that may not be positioned in the tropics, but have a lot of production in the tropics and are looking for somewhere where you have US law to protect a lot of your intellectual property. So we've been able to pull in funding that way as well. But we also submit grants to, all competitive sources. So a lot of this coffee work is being done on a USDA SCRI grant. I just submitted for that proximal sensing and metabolomics work, a grant to NSF with Dr. Michael Gore at Cornell. So we can kind of go a lot of different routes for funding.
For the coffee leaf rust, are you also looking at varieties bred elsewhere with resistance and
Yes.
seeing how they do?
Yes, yeah. So within those farmer trials and within actually every bioassay that we've run, we have positive and negative controls. So we're using both the local types for our control to make sure that our inoculations are actually have the potential to affect. And then we also have Obatas from Brazil and different Catimor hybrids that we're using to see if the resistance is there. Yeah.
Oh, is there another part? I'm
Oh,
sorry.
okay. I'm sorry, I'm not repeating the questions.
Yeah, it's all right. You're close enough to the mic, hopefully. When it comes to the population improvement side of breeding versus product development, once you get through these initial germplasm characterizations with your base populations, do you think if you replicate those population improvement groups and genotypes in different production environments with your partners, you'll be able to shorten that cycle time that you found with your modeling?
Potentially, yes. Yeah, I think it depends on the crop type for sure. But being able to, especially if we have farming partners that might be able to take advantage of a environment that we might not have, that would be extremely important in terms of just trait accuracy, and understanding environmental impact of those traits. What I'm really interested in just at this stage is just population improvement, and then variety development is a whole nother animal, and understanding with moringa, we may want to have more diversity in an orchard because of how frequently they're cutting it back and how long it's going to be in the field. Could see disease as being a potential issue in that type of a circumstance. So having that variation there would be important. Other crops that are rather quick, you might be able to get away with, you know, a biparental cross for variety. So it's circumstantial, I would say.
And then real quick, with my prior breeding programs I managed, a big struggle was when doing neo-domestication, while simultaneously rapidly improving genetics, you have rapidly shifting agronomic practices.
Mm-hmm.
How do you foresee balancing that across these different crops where it might be entirely new market? So the tools to produce are going to shift pretty quickly on you.
The way I think about it is most of the world is either mechanized or is moving towards mechanized. So taking that into consideration is important. I know that if you have probably smallholder farmers where a lot of it is just hand labor, that situation might be different. But yeah, how to balance that is, and how to predict that is difficult when you're thinking this long term. Yeah.
Any last questions?
Just a quick technical question.
Yes.
I think a limitation of alpha Simar, to my understanding, is you can only have 2 alleles per locus, so you
Correct.
cannot have multiple alleles.
Correct. Yeah.
So have you thought about what are the implications of that?
Yes, I have, and it's one of the limitations that was difficult to grasp with, especially even considering not being really reliably able to track dominance and epistatic effects within this is something else that it's, you know, that I meant to put in a limitation slide into this presentation, but there's a long list of limitations with simulations. So grounding that and understanding that is important. And that was an oversight on my end. I should have included a limitation slide. But there's a long list, and there's hundreds of other parameters that could be considered within this as well. So if anybody wanted to take this and run with it, there's a lot of opportunity. Yeah, absolutely.
I worked in the tropics, and it looks like soil management could be an issue there, and soil fertility, pH management. Have you found that to be a problem?
In Hawaii, yes, we have, especially in Monowili, our pH on a good plot is 4.8, and a bad plot is probably 4.1. The structure is great, but the chemical composition of the soil is just miserable. So when we plant, we'll put anywhere from 2 to 4 tons of lime per acre, if not more. And
It's a volcanic soil.
yes, very much so. Yeah, so it is tricky. So we take advantage of both composting as well as low release fertilizers, and that's a huge part of Johanni's work. She's a soil scientist, so she's very much trying to figure out how we can improve our farm soils. Yeah, because it is tricky.
With that, folks, we'll wrap up. Nathan is here for the rest of the day. We did have some openings in his schedule as well, so if any staff or students would like to meet with him, please reach out. Otherwise, thank you, Dr. Fumia, for the presentation.